Dear students, last time we discussed about the structure of the gills, and today we will be continuing with the respiration in fishes. In this part, we shall be dealing primarily on the mechanism of respiration. Now, we shall also be dealing with the vascular supply as well as the gill area and mechanism of respiration. Let us have a quick recap of what we discussed last time. We knew that elasmobranchs do not have gill covering, but they have gill slits. Herein, these fishes, that is the osteichthyes, bony fishes, they have the gill slits, but the gill slits are covered by an operculum. And when we talk about these very sharks, they have the spiracle, whereas the spiracle is absent in the case of bony fishes. And this is the structure wherein last time we discussed that the sharks have normally 5 pairs of the gill slits, whereas the bony fishes have the 4 pairs. But in the case of elasmobranchs or the chondrichthyes, they can range from 5 to 7 pairs, whereas in the case of bony fishes, the gill slits never exceed 5 pairs. So this is the structure. These are the gills in the case of the bony fish. These are the internal arrangement, and these gills are showing when they are taken out. And simply, this is the head side. This is the head side of the fish. This is the tail side of the fish. So toward the head side is the gill arch, wherein the gill rakers do face toward the head side, whereas the gill filaments face toward the tail side. And in some fishes, a pseudobranch is also present, which is a false gill. Now, last time we also discussed about the gill rakers, that the gill rakers vary according to the feeding activity as well as the type of the fish species. The gill rakers are strong in the case of carnivorous fishes, whereas they are very much filamentous in the case of planktivorous fishes. These are the gill rakers. These are the gill rakers teeth-like in the case of in these omnivorous fish, Serranus megala, whereas in the case of planktivorous fish, the gill rakers, they are the filamentous. And when we take apart, when we take, for example, this only one filament, only one, you can say, part along with the gill raker, it looks like this. For example, this is called as the holobranch. This is called as the holobranch. That is the complete one gill. So this very gill consists of the gill raker toward the inner side, whereas the gill filaments toward the outer side. Now there is one confusion among the students, that what are the gill filaments, what are primary gill lamellae, and what are the secondary gill lamellae. Now, to avoid this very confusion, keep in mind that this the things which we can see, which we are able to see, for example, I may be showing you, for example, this is the part which we are able to see. These are the gill filaments, are primary gill lamellae. So these are primary gill lamellae. This is a primary gill lamellae here, and this is a primary gill lamellae. But these very creases on the primary gill lamellae, which increase the surface area. For example, here in this very case, I am going to show you by the red color that, for example, these very creases, which are like this crease, like this crease, like this crease, which increase the surface area of the gill filament or primary gill. It is the secondary gill lamellae now, and this very part is called as the interbranchial septum. This very interbranchial septum is also varies. For example, here in this very case, elasobranchi, it is rightly up to the more than the length of the gill filament, whereas in the case of holocephaly, it is almost equal to that of the gill filament. And here in the case of holostri, it is about half of the size of the gill filament, whereas in the case of teleost fishes, then they are very short, and they are very short. So let us see, let us have a discussion, a little bit discussion on the vascular supply in the case of teleostean gill. Here what we are seeing that these are the, I will show you that these are the gill arches. These are the gill arches. 1, 2, 3, 4, and 5 gill arches. And this is the buccal cavity here. So this is the operculum. So these are gills are inside the operculum. When we are taking out one gill arch, for example this one gill arch we are going to take out, we are enlarging it, it looks us like this. Here in this very case, now the gill rakers, these are the gill rakers, these are gill rakers, these are looking as teeth like strong. So this is surely a carnivorous fish, because the gill rakers are very strong, and comb like, strong teeth like, they are in the case of carnivorous fishes. And these are the gill filaments, we call them as the lamellae or primary gill lamellae. So this is a little bit more enlarged view. So these are the creases on the gill filament, these are the creases on the gill filament. So these are the secondary gill lamellae. So again it is enlarged here. In this very case it is enlarged. I am going to show you. For example, in this very part, this is the one, for example we are taking this very filament out, and I am going to show you here in enlarged view. So what we are seeing, this is a one gill filament, it's a primary gill filament, and on this very primary gill filament there are creases which emerge out of the primary gill lamellae. So these are called as secondary gill lamellae. So these are the creases, secondary gill lamellae, they are highly vascularized. So exchange of gases takes place inside the secondary gill lamellae. This is the important thing, that these are highly vascularized, and the exchange of gases takes place by the network of the capillaries, wherein the oxygen poor blood. So this is the oxygen poor blood which is coming out of this very afferent artery, and then it is moving on to the efferent artery. So here in the exchange is taking place here, exchange is taking place here in this very part, where the flow of the blood is in this very direction, flow of the blood is in this very direction, and flow of the water here, I have shown that the flow of the water is in this very direction. So when the flow of the direction of the blood and the flow of the direction of the water, they are in the opposite side, we call this when the this flow of the blood and the flow of the water, it is in the opposite direction, we call this very mechanism as the countercurrent mechanism, so that the maximum exchange of the gases takes place. So we all know that the water has the dissolved oxygen in the form of the as a dissolved oxygen. So the blood which comes out through the afferent artery and leaves through the efferent artery. So it comes in contact with the oxygen rich water. So exchange of the gases takes place. The oxygen from the water gets into the oxygen deficient blood, and the carbon dioxide from the oxygen deficient blood goes to the water, and the water leaves out of the gills, and thus the exchange of gases takes place. This is a summarized way. Now we shall be talking at length about this very in the next slide. Now, first of all, we shall be talking about because we should be having an idea that what is the vascular pathways? What are the vascular pathways which are found in the gills of the certain teleost? There are two vascular pathways. One is respiratory pathway, and other is non-respiratory pathway. So, respiratory pathway is associated with respiration, whereas non-respiratory pathway is associated with, now you have to keep in mind that the non-respiratory pathway is associated with the nutrition and the circulation of the hormones, particularly the circulation of the hormones and the nutrition. We shall be discussing this with the help of the. So there are two vascular pathways in a gill. One is respiratory pathway and other is non-respiratory pathway. Respiratory pathway is associated with the exchange of gases or with the respiration, whereas non-respiratory pathway is associated with nutrition and circulation of the hormones. Let us have a little bit of discussion that how these very two pathways work. So the first of all, we are concerned with the afferent branchial vessel. I am showing this very oxygen deficient blood with the help of the blue and oxygen rich blood with the help of the red. So I'm using this very blue to demarcate that this very afferent branchial vessel is associated with the oxygen less or oxygen poor blood. So this very afferent branchial vessel brings oxygen deficient blood to the gill, and it runs through the entire length of the gill arch and gives off the number of the primary afferent branches. So it gives number of the primary afferent gill branches. So to every gill filament, to every primary gill filament, it gives off a branch called as the primary afferent branch, or primary filamental arteries to the primary gill lamellae. Each primary gill lamellae or the primary afferent vessel. Now this very primary afferent vessel divides laterally into number of secondary vessel. These very primary afferent vessels divide into the number of the secondary. These are the secondary gill lamellae, from the primary gill filaments to the secondary gill filaments, and from the primary afferent vessel to the secondary afferent secondary vessels. So one each for secondary gill lamellae. These run across the gill rays, dividing again into 2 to 4 tertiary branches. So these very secondary afferent vessels divide into 1, 2, 3 and 4 tertiary branches. So these are having the oxygen poor blood. So in the secondary lamellae, these capillaries break to form lamellar channels interconnected with each other, forming a vascular core, and thus these form a vascular core of the secondary lamellae. Exchange of gases takes place by the flow of the blood in these channels. So exchange of the blood actually takes here in these very channels. The oxygenated blood is collected by, now I'm going to show you by the red, that this very oxygenated blood is collected by the efferent lamellar arterioles, which carry blood to the primary efferent vessels. So this very part, half of the part is associated with the oxygen poor blood, having the oxygen poor blood, and this very part is having the oxygen rich blood, now which carry blood to the primary efferent vessels running along the margin of the gill lamellae. So along the margin of the gill lamellae, so blood is finally carried to the, so these from these very vascular to the secondary to the primary efferent vessel, and then this primary leaves via efferent branchial vessel. So half of the portion deals with the oxygen poor blood, whereas half of the portion deals with the oxygen rich blood. Now we do have another this very part which is associated with the second one, that is the non-respiratory pathway. It consists of the complex arrangement of this sinuses and veins that carry blood direct to the heart, bypassing the systemic circulation. So it carries blood directly to the heart without going to the tissues, without going to other body parts. So it bypasses the systemic circulation. So it is believed that only function of this very pathway is the provision of the nutrition. It is associated with the circulation of hormones and the nutritive blood channel, and the central nutritive. It is a nutritive blood channel, and it deals with the circulation of the hormones. So we have discussed this very pathway. Now again here in another this very diagram. So this is the oxygen poor. This is the oxygen rich part. The structure of the teleost gill is of the type that the water is brought close to the secondary gill lamellae, where exchange of the secondary gill lamellae, close to this very secondary gill lamellae, and exchange of the gases takes place here. So gills utilize almost 50 to 80% of the oxygen here. 50 to 80% oxygen present in water is utilized here, and the teleostean gill is supposed to be more efficient than the elasmobranch gill due to the great reduction in the interbranchial septum. As in the earlier part of this very lecture, I told you that the septum in the case of elasmobranch is very large, and in the case of teleost it is very much reduced. So it is more efficient than that of the elasmobranchs. Now again, when I was talking about that this very part, that is the oxygen poor blood comes via this direction, and the water flow, and the water which is having oxygen rich, it comes in the opposite direction. So this very oxygen poor blood comes in contact with the oxygen rich water. So this is called as the countercurrent water flow, and it makes it more efficient for the exchange of the gases. Whereas in case of the tench, that is tinka, the countercurrent mechanism is so efficient that 51% of the oxygen from the incoming respiratory water diffuses in, and 9% of the outgoing 9% of the oxygen from the outgoing water currents. So um, the water brings along with it the dissolved oxygen, whereas the blood brings along with it the blood which is having poor in oxygen. So the exchange of the gases takes place by this very countercurrent mechanism. Now, what is the mechanism of the respiration? We have talked, yes, the exchange of gases takes place in the secondary gill lamellae. But how what is the mechanism behind this very respiration? Now, for this, we shall be having a little bit idea, and that we knew that this is the mouth, these are the gills, the heart is associated with it. Now in the case of fish, there is only single circulation. What happens in this very circulation, that impure blood or oxygen deficient blood comes directly to the gills, and then this very oxygen rich blood goes to the heart, and then to the different parts of the body. So it is a kind of single circulation. So in this very part, we have to keep in mind that the opening and the closing of the mouth, and the opening and the closing of the opercula plays an important role in the respiration of the fish. The continuous undulational flow of the water over the gill surface is accomplished by the respiratory pump. Now, I have used the word pump, respiratory pump. Now, what is this very respiratory pump, and what are the organs included in this very respiratory pump? The respiratory pump of a teleost consists of, now you have to keep in mind that this very pump consists of the buccal cavity, the first one, that is the buccal cavity, and two opercular cavities. Now this mouth and the opercular cavities, these comprise the respiratory pump. So the cause, it is caused by the movement. Now how this very respiratory pump functions. Now it is the movement of the opercular, you can say, bone, opercular cavity and the bone, and the mouth, which functions the which helps in the functioning of this very respiratory pump. In the beginning, what happens? In the beginning, what happens? The water enters into the mouth by the expansion of the buccal cavity. Now, when the buccal cavity is opened, this very cavity expands. So water is then accelerated over the gills by the simultaneous contraction of the buccal cavity, and the cavity contracts, expelling the water out through the opercular opening, and the cycle begins again. So what happens? The water gets in via mouth, then the mouth is shut, and the opercular bone is still, opercular wall is still shut, the pressure is created, then the water leaves. When this very opercular bone or the wall is released, it is opened, the water leaves, and it goes through the gill filaments. It goes on by passing in between the gill filaments, and particularly the primary gill filament and the secondary gill filament. So the water goes enters via the buccal cavity and leaves via the gills. Now here is the function of the gill raker, what I was telling you. Here it is the function of the gill raker, that when the water comes in, the it is filtered through the gill rakers. It is filtered through the gill rakers, and then it leaves out. So by filtering the water, the planktons and the food particles, they get stuck in the gill rakers, and then they are passed onto the alimentary canal, whereas the water leaves out, and it has respiratory function as well as the function in the digestion as well, because it helps in the capture of the food particles, whereas its second function is that it protects the gill filaments, which are very much delicate. Let us have a look how this very mouth opens and shuts. This is the mouth. This is the mouth. This is the oral wall, and this is the mandibular arch. These are the gill rakers on this very side. These are the gill filaments on this very side, and this is the gill arch. This is the gill arch, and then this is the branchial aperture. This is guarded by a wall, and then these very gills are strengthened by the bone called as the branchiostegal rays. These are called as the branchiostegal rays. So so on one hand, this is an open mouth, and on the other hand, this is a closed mouth. Here in this very case, the mouth is open and the gill apertures are closed, and in this very case, the mouth is closed and the gill apertures are open. So this is the inspiration. This is the inspiration, and this is the diagrammatic sketch wherein it shows the expiration. Now, this is the actual mechanism what we are going to study now, and dear students, you have to keep a vigil on this. I will tell you the 4 steps what happens during the mechanism of the exchange of the gases. Now we have noticed in our earlier diagram that what happens when the mouth is open and what happens when the operculum is closed and then open. Once the mouth is open and it is inspiration, and another time these very opercular valve is open, it is expiration. But what happens in between, we have to talk about this. So for respiration, the mouth is opened, buccal cavity is enlarged. So this is the oral valve. This is the oral valve, and this is the buccal cavity. Now this is the buccal cavity. Here this is a diagrammatic sketch wherein we have to show the pressure inside, the negative pressure and the positive pressure, and then how this very exchange of gases takes place. When the mouth is opened, buccal cavity is enlarged and the branchiostegal rays are spread and lowered. Thus the buccal cavity increases. And creates a negative pressure. Now what we are seeing here, that a negative pressure is created inside the buccal cavity, and the water is sucked in, and thus the water is sucked in. When there is a negative pressure in the oral, you can say buccal cavity, the negative pressure is created and water is sucked in. This is the one step. So when this oral cavity is filled with water, now when the water gets sucked in, oral cavity is filled with water, and the mouth is closed and the operculum is moved anteriorly to increase the opercular cavity. Now when the mouth is full of water, this very operculum is a little bit moved, and the cavity increases. This very opercular cavity increases. And what happens with this increase in the opercular cavity? But opercular cavity is increased, but the opercular, this I'm showing you that this is still closed, that the opercular valve is still closed, and thus negative pressure is created inside the opercular cavity. So due to the pressure of the so what happens when the oral cavity is filled with water, mouth is closed, and the operculum is moved anteriorly to increase the opercular cavity. But the opercular opening is closed due to the pressure of the external water. So there is a pressure from the external water as well. A low pressure is thus created. What I was telling you, that the low pressure is created here, which is very much low. Here it is shown by the 3 negative minus. So low pressure is created in the opercular cavity, and water flows over the gills into it. Now from this very buccal cavity, the water flows over the gills inside the opercular cavity into it. So what happens next now? So the buccal cavity and the upper cavities are reduced to exercise the pressure of the water. So now they are reduced to exercise the pressure of the water inside it. The oral walls prevent the water from going out of the mouth. So what happens? The oral wall is closed now. It is closed so that the water is not so water doesn't leave out. The oral walls prevent the water from going out of the mouth. The opercula, after reaching maximum abduction, so when it reaches the maximum abduction, are quickly brought towards the body. So water is expelled through the external branchial aperture. Now here what we are seeing now, the water is expelled through the external branchial aperture and is prevented from going back due to the excess pressure. Now as the pressure is increased inside the buccal cavity as well as the opercular cavity, the water is now leaving outside toward the outer side, as compared to as the pressure is excess in the buccal cavity as compared to the opercular cavity. So now what happens here, that the opercular valve opens, it leaves out, and the mouth again tries to close, and then again negative pressure is created. So this very cycle goes on. We call it as the respiratory pump. This is a diagram by which I want to show you the countercurrent mechanism, which I discussed in the earlier slide, that how the countercurrent mechanism works. I was telling you about that the exchange of gases takes place in the secondary gill filament. For example, here in this very case, the here that this is the primary gill filament on which there are the creases to increase the surface area, and these are the secondary gill lamellae. So this is the primary gill filament, and the secondary gill filament are the primary gill lamellae or the secondary gill lamellae. So these are the creases. Wherein there is one interesting thing, that the water rich in the oxygen comes from this very side toward this very side, and the blood deficient in oxygen moves from this very side to this very side. So herein I have tried to show that by the help of this very arrow, that this is the movement of the water and this is the movement of the blood. So what is happening in this very case? For example, the water is coming with the saturation of the oxygen. For example, here I have shown it by the 100, 90, 80, 70, 60. So it came with the 100% you can see oxygen, or it is saturated with the oxygen. It is having the 100 oxygen while moving toward the gills, through the secondary gills, and the blood is exchanged. The movement of the blood is from this very direction. From this very direction, the movement of the blood is from this very direction to this very direction. So this is the direction of the water. So what is happening here, that the blood which is having less oxygen, it is that, for example 50 here, it is coming in contact with the water which is 100 here. So there is a loss of the oxygen from 100 to 50, whereas there is increase in the oxygen content in the blood from the 50 to 90, almost, for example 50 to 90, almost 40% increase, or the 40 increase in the you can say saturation of the oxygen in the blood. So on one hand, what we are seeing here, that the water is flowing on this very direction, whereas blood is flowing from this very direction, and thus this very coming in contact of the blood and the oxygen, the maximum exchange of the gases takes place. Now one important, this is called the countercurrent mechanism. Now one important thing what I want to tell you is, again, it is the gill area. Gill area. Now, I want to just make you aware that the relative number and the size of the gill lamellae determines the respiratory area of the gill of a fish. So, number and the size of the gill lamellae, so they determine the respiratory area or the gill area of the gill in a fish. So, the total gill area of a fish species is calculated as a product of the total length of the primary lamellae, primary filament, and the frequency of the secondary lamellae. Now, what is gill area actually? It is actually the length of the primary gill lamellae, and the frequency, that how frequently the secondary lamellae are present in the on the primary gill lamellae. So together all this comprises the gill area. Now again, I was talking about that this very gill rakers are also dependent species to differs from species to species. Similarly, the gill area also differ from species to species. Some have the long gill filament, some have the short gill filament, depending upon the activity of the fish. Actually, it is actually the activity of the fish. The more active the fish, more the gill area, because an active fish requires more oxygen than that of the sedentary fish. So fish which is not so active, that has the lesser gill area than that of the fish which is very much active. Now another important mm, mechanism is seen in the case of, particularly in the case of elasmobranchs, and that is ram ventilation. Now most sharks rely on ram ventilation, forcing water into the mouth. So they forcefully take the water into the mouth and over the gills by rapidly swimming forward. So they move forward and gulp the water forcefully. So this is the ram ventilation. This is a ram ventilation. So in slow moving or the bottom dwelling species, especially among the skates, as we all know that these are the bottom dwelling and little bit sluggish, and the rays which are sluggish, a little bit not so active as that of the other fishes. The spiracle is enlarged in them, and the fish breathes by sucking water through this very opening. So this is the role of the spiracle, that this spiracle tends to suck the water and helps in the breathing by sucking water through the opening instead through the mouth, in the case of the fishes which are less active. Whereas in the case of sharks, they move fastly and they can gulp the water forcefully, but in the case of sluggish fishes, they cannot forcefully gulp the water because they are slow moving, and hence here the spiracle makes a role. Now another one important thing is that the interruption, when there is interruption in this very ram ventilation, there occurs the cough. So cough is the reverse of the ventilation. So ventilation is the sucking of the water, whereas cough is pushing the water out. Now here the cuff is also important. It is not that the cuff is not important. Cuff is also important. For example, when the gill rakers get entangled by the help of the impurities or the foreign matter, the fish forcefully cuffs. It is in the case of teleost well as in the case of other fishes, that they forcefully cuff, so that as to clear the excess mucus from the gills, and to clear the excess foreign substances from the gills. So these are certain important things, that the operculum is also plays an important role in adjusting the pressure of the water inside the pharynx, as we have studied by looking at the diagram as well, that how negative pressure is created, how positive pressure is created, and how buccal cavity is closed, and then pressure is created in the opercular cavity, and how the opercular cavity is then wall is released and the water leaves out because of having the higher pressure inside. So we come to know that the operculum is very much important in adjusting the pressure of the water inside the pharynx as well. So walls inside the mouth keep the water from escaping. So the walls are present in the mouth, as well as the walls are present in the opercular cavity as well. So slow moving, we have discussed that the slow moving bottom dwelling fishes, they have the spherical instead of the forceful grasping of the water. Instead they use spherical for this sucking of the water. Now it is worth to mention here that there is certain variation in the respiration pattern in this because there is a pattern of respiration different in the case of fast swimming species, in the case of bottom dwelling species, and in the case of hill stream fishes. For example, in the case of fast swimming species such as mackerels, the salmons, the mouth and the gill flaps remain open and bathe the gills with water current. So their operculum remains open. So from the mouth, water enters, from the operculum it leaves out during the swimming, and this very exchange of gases takes place. Whereas in the case of the fast swimmers, there are small cavities, whereas in comparison to that of the sedentary fishes. Now, in the case of bottom-dwelling fishes such as flounders, Pleuronectidae, goose fishes, flatfishes, eagle rays, stingrays, and they possess enlarged dilatable opercular cavities. Their mouth does not open widely, and so during inspiration, causing slow and deep breathing movements. So they have the slow and deep breathing movements. And the other bottom dweller species such as morays or eels, morinidae, they keep mouth open during the respiration. And in some fishes such as bonefishes and the goldfishes, when they find prey covered by the sand grains, they release jet of the water from the mouth with the help of the strong adduction of the gill covers, and they uncover their food or prey. So this is a kind of the forceful pushing of the water, so as to remove the sand particles from their prey. In the case of hill stream fishes, such as because hill stream fishes, they have one problem, that the flow of the water is very speed, and such as hill stream catfishes, they have to attach their body temporarily to the substratum. The water intake is accomplished by developing grooves protected by the barbels, with slight reduction in the force of the suction. So ventilation is carried out only by the opercular movement. So it is the control of the opercular movement, the respiration or the ventilation of the gills takes place. In the case of certain other fishes, while attaching to the substratum with their suctorial mouth, because we all know that the certain hill stream fishes, they are suctorial, because they have to attach themselves to the stones or pebbles, otherwise they will be swept away. So the larva of the for example, the larva of the South American lungfish, that is Lepidosiren, and the reed fishes, they have the external gills in that very case. They have the external gills. These very fishes have adapted themselves according to the different environments, and they have modified themselves to suit their particular environment.
So this was all about the respiration in the fishes, the mechanism of respiration. Thank you very much.